Tissue culture. Tissue culture is one of the process in which the endangered and extinct variety of the rare species can be produced by the various method. It is one of the important branch of biology nowadays because most of the plants are going to be extinct. So earlier I have discussed about the anthroculture, micropropagation, and also the totipotency. Today we are discussing about the protoplast culture. Protoplast culture is also known as somatic hybridization, and the protoplast of the different species or the genera can be fused to produce the somatic hybridization. Tissue of the two different plant species are taken and first of all the cells are separated and the cell wall is removed. The cell wall is dissolved or treated with the help of the certain enzyme, and that enzyme is known as the peptidase and cellulase enzyme. These enzymes separate the cell wall and only the protoplast is left, and that is known as the naked protoplast. These two naked protoplasts of the two different species are taken in the culture media and they are fused. Basically, the fusion of these two varieties of the protoplast on the culture media is done at high voltage electric current, or the enzyme that is known as the PEG and the sodium nitrate. The full form of the PEG is the polyethylene glycol. Both the cytoplasm of the nucleus are fused together and the hybrid protoplasm is produced. These hybrid protoplast is placed on another culture media for the further growth, and if the fused protoplast is transferred to the other media, the new cell wall is developed. They are called somatic hybridization cell. The hybrid cells give rise to the callus. The callus later produce the embryoid. So what is embryoid? The embryoid is the embryo-like structure. Embryo is known as the embryo that develop from the zygote, but the embryoid that develop from the callus. Most of the plant species are cultured to develop the hybrid protoplast or the hybrid somatic cells. First somatic hybrid plant was obtained between the two species of the tobacco, that is the Nicotiana glauca and the Nicotiana longsdorffi. It was done by the Carlston et al. 1972. Pomato is the somatic hybridization. Between the two plants, that is the potato and the tomato. They belong to the two different genera but belong to the same family. After the somatic fusion, the same plant having the tuber and the fruit of the tomato, and the several other experiment was done. Then, such as the rice, the wheat, maize, brassica, etc. The another somatic hybrid was produced between the rice and the carrot. The plant having the carrot and the rice. So it is one of the important method. Here is the importance of the protoplast culture. It helps in the development of hybrids, asexually produced reproducing plant, possibility for the development of the new crop plant. So for example, the tomato help in the genetic manipulation, male sterility, and the hybrids are produced. Mutation is easier in this plant. Here is the diagram that is the given between the two plants. Here is the Nicotiana glauca having the chromosome number 2n is equal to 24, and Nicotiana longsdorffi is equal to 2n is equal to 16, the free cells are taken, then the cell wall is removed with the help of the enzyme, and then these two, the naked protoplast are fused, the culture media and the somatic hybrid is produced, and then it is transferred to the another media which we have already discussed, and finally the plantlet is produced. Thank you.